I want to call your attention to two things that are on page 247, which I actually highlighted. So in the second paragraph, it mentions that some intertidal organisms don't use the so-called run and hide or the clamming up approach to prevent themselves from drying out. So what the book has said prior to that is that some animals will just hide, they may burrow into the sand or something like that, or some of them will clam up. That's where the expression comes from, that the clam will draw its shells together, the two shells together, and that will actually maintain moisture. And there are problems associated with that too, because if you're clamming up, you can't feed very effectively. You can't feed at all while you're doing that. So that's kind of not necessarily the greatest strategy. But right after that, it says some intertidal organisms don't do that. Instead, they simply tolerate drying out. And it mentions something that I found quite remarkable. It says some intertidal chitons can survive the loss of 75% of the water in their tissues. And then there are some varieties of rockweed, fucus being the example I think that they actually give here, that can actually lose 90% of their water, to the point where they actually become sort of almost crumbly. Nevertheless, if these organisms are put back again into water, they rehydrate again, and they don't seem any worse for wear. They don't die. So I mean, while they're in that dehydrated state, however, they're more vulnerable to breakage. But as long as they can remain kind of in stasis, and until the tide comes back in, it's always possible for them to rehydrate, and they shouldn't suffer any really dire consequences as a result of that. So the question that I actually wrote under here is, how are organisms such as intertidal chitons and fucus able to withstand desiccation? What do they actually do that enables them to withstand desiccation? Because that's really quite remarkable, to lose 3/4 or as much as 90% of your water and still be able to survive. And then the other thing that also drew my attention was in the paragraph on temperature and salinity, where it says certain species of periwinkle from high in the intertidal show remarkable heat tolerance, and one is known to survive temperatures as high as 49 degrees Celsius, 120 degrees Fahrenheit in the laboratory. So that's really a pretty remarkable thing as well. And both of these things, there's some relationship to being able to withstand changes in temperature and salinity, and also to be able to withstand desiccation. So I want to talk a little bit about the chemistry of how the chemistry, and to some extent the physics of how this actually works. So it turns out that there is a molecule. I'm going to show you the structure of the molecule. It's a disaccharide that is two sugar molecules that are put together, and it's called trehalose, trehalose. And trehalose is as I say, is a dimer of. See if I can be able to see this. The lighting in here is not great, but this molecule. Let's see if I move it back like that. Let me see if I can illuminate this a little bit better. So what you see there are two 6-membered rings here with oxygens in them, and that's actually these are called pyranoses. The 6-membered ring is called pyrane, and these pyranoses or pyranoses, they're sometimes pronounced. These are sugars, and this particular sugar is very interesting because it has some unusual properties that we don't associate. This is actually made in animals. Animals store their sugars in the form of glycogen, and there is an enzyme that will actually cleave glycogen to produce trehalose. Now, trehalose seems to be made when animals are under a certain amount of stress. The machinery for producing trehalose is really turned on full force. So trehalose itself has high water retention, and it actually forms a gel phase when it comes in contact with water. So as cells dehydrate, this gel phase is formed. And what this does is it actually prevents disruption of the organelles within the cells. And it does so by actually sort of splitting them into position, because the organelles are quite sensitive, and if you just pulled the water out of the cell, all the organelles would kind of mush together, and if you tried to rehydrate them again, you just get a mess. But by actually splitting these things, by these molecules acting as kind of intermolecular splints, they actually hold the organelles in place. So then, when rehydration occurs, it's not lethal to the organism. And normally, rehydration of a completely dehydrated organism that doesn't have this kind of this kind of a protective molecule, that would be fatal. So the other thing about trehalose is it also has antioxidant properties, and that seems to be also important for preventing things from drying out. So trehalose is found in a lot of different creatures, but particularly you would find it in chitins, and you'd find it in a lot of other organisms that undergo this kind of drying out process. So there are a couple things, a couple other organisms that dry out almost completely. One of them, there are organisms that are called tardigrades. Now these things are actually, they're aquatic, but they're fresh water, and they're sometimes called water bears. And when they dry out, their bodies convert all the glucose that's present to trehalose, and then they go into a resting state where they actually appear as though they're dead. But and this particular state is called cryptobiosis. But once they are rehydrated, that is, once water is put back in, they actually spring back to life again. So there are a number of organisms that do this kind of thing. There's another thing that years ago they used to advertise these things in comic books. They were called sea monkeys. You probably still see them around today, but they'd have these. They would tell you that, you know, if you send in a certain amount of money, they would send you a kit that had these cute little sea monkeys. Well, they don't look at all like monkeys, and they're not really very cute. But what they are brine shrimp. Artemia is the genus, and the Artemia, when you get them, they're almost. I don't think they're really eggs. I think they're almost encysted in some form. So they actually have a sort of a coating around them. And if you put them into water, the cysts will actually the animals will actually develop out of the cyst, and they'll actually spring to life. And that usually takes 24 hours from the time that these things are placed into salt water. So it's another example, and I believe you can get them to re-encysted again by just removing the water, and you probably can reverse this process a number of times with Artemia. So these are just a couple organisms that do this kind of thing. And fucus probably does something similar to that, although fucus actually also secretes a mucilage, kind of a sticky substance that actually, to some extent, retards or slows down desiccation. But once desiccation actually does occur, it doesn't seem to hurt the fucus at all. So not every variety of fucus is very desiccation tolerant. There are some varieties that live in brackish water. That's the water that's not really very strongly salt water. And those particular organisms, those particular forms of fucus, they will dry out, and they don't really have this so much of an ability to snap back once they're rehydrated again. But the ones that actually live in salt water, which tend to be much more resistant, they've got, what happens is they actually have a lower respiratory rate than the ones that live in brackish water, and they also have a higher photosynthetic rate. So they're actually they undergo photosynthesis much more than the brackish organisms. And finally, they have a higher concentration of mannitol, another sugar. And although I can't find direct evidence for this, I sort of suspect that the mannitol is another one of these molecules that's actually acting as a cryoprotectant. That is, it's acting kind of as a maybe as a an antifreeze, and it probably is also protecting the organism from any deleterious effect as it starts to dry out. It's preventing it from falling apart. So with fucus, that's probably what's happening. In terms of these other organisms, or at least in terms of the organisms that are using trehalose, and I don't know whether fucus also may be using trehalose. I'm not really sure. But trehalose, this molecule that I'm showing you here, along with another molecule, glycerol, which is a much simpler molecule. Glycerol is just a glycerol actually looks very much. I'm going to erase the trehalose here so I can put in the structure of glycerol. But glycerol looks very much like antifreeze, ethylene glycol. So the difference between ethylene glycol, which is the material that I've drawn on the top there, this OH, CH2, CH2OH, that's ethylene glycol. That's what you find in your car's radiator as antifreeze, antifreeze, and this other molecule here is glucose, which you'll notice has just an extra carbon in the middle that has a CH and an OH attached to it. That has a hydrogen and a hydroxy group attached to the carbon. So it's CHOH, CH2OH, OH, CH2, CHOH, CH2OH. And that used to be used actually as an antifreeze. But that is a naturally occurring antifreeze, and it's something that you'll actually find associated with trehalose in a lot of the organisms that are using trehalose. Not only are they using the trehalose to prevent desiccation from destroying their integrity, but they're also using the glycerol to prevent damage to intracellular damage that would occur once the temperature drops below freezing. So another function of the trehalose, by the way, is that it is shown to stabilize and renature inorganic pyrophosphatase. Now, pyrophosphatase is an enzyme that actually converts pyrophosphate, which is P2O7, pyrophosphate in the presence. This is what actually happens. For instance, if you're going to cleave ATP and you cleave it, if you actually took off two phosphate groups, that would be a pyrophosphate. If you take off one, it would be a single phosphate, but the pyrophosphate is two phosphates linked together, P2O7, and in the presence of water, that can be converted into two phosphate groups. But it does require this enzyme called inorganic pyrophosphatase. So another function of trehalose is that it actually stabilizes that enzyme, and should that enzyme actually become denatured because it's been dehydrated or it's been subject to too much heat or possibly some kind of physical trauma, that it's actually possible for in the presence of trehalose that will actually renature the enzyme and cause the enzyme to start functioning again. So this is another way that trehalose actually serves as a kind of protectant. It's also called a molecular chaperone because of this particular property that it has. Molecular chaperones, or these chaperone molecules, what they do is they assist other molecules, particularly things like proteins, that may become misfolded in some way. It enables them to fold properly, to actually have the proper tertiary structure, so that when a protein is subjected to too much heat or pH changes, it can actually lose its native folding, and that inactivates it. And these kinds of molecules, these molecular chaperones, enable the molecule to fold properly so that it has the correct structure to carry out catalytic activity.